Institute in different places, mostly in the Maritimes, but a little bit in other parts of the country and internationally. So I'm just going to go through this, and then there'll be time at the end for questions, and I'd be more than happy to entertain those. It's also a little weird for me because I usually like to make jokes, but I don't have any audience in front of me except Michelle, and Michelle doesn't seem to like my and appreciate my jokes as much as she used to, so I'll do the best I can. Right. So the research focus in my lab, and what sort of, I guess, best explains the kind of work that I prefer to do, is really very much field-based. So I spend most of my time, or at least my students spend most of their time, focusing on questions that are answered by doing a fair amount of field work. We do tend to supplement that with some laboratory-based work, but it's largely field-based, very much an applied science emphasis. And wild Atlantic salmon are really very much the study animal of choice for me. Michelle often accuses me of being salmocentric, and I don't apologize for that. I think it's a marvelous animal to be looking at, and it helps us understand an awful lot of things, especially about rivers, which is where I like to spend most of my time. So there's three main areas of focus of my research that I'll be covering today. The first one is natural stressors, and what I mean by that are sort of natural impacts, things that occur naturally. Winter, for instance, how ice affects things, flooding events, naturally occurring, not so much anthropogenically induced floods, but floods that are sort of typical based on the sort of hydrologic regimes in our rivers. And again, high temperature is another one of these, and this has become much more important in recent years, especially with the number of the climate change scenarios that are suggesting we're likely to have higher summer temperatures in the future, which will have implications for salmon, and I'll talk about that in a little while. The other thing that a second area of focus for my research program is human-induced impacts, and forestry has been one I've looked at a little bit in the past through the project up at Catamaran Brook, where I do a lot of research in the Miramichi, and also more recently hydroelectric operations and how they affect so fishes in rivers, and specifically Atlantic salmon in any of our rivers. And finally, sort of a broader, more all-encompassing area of research is how ecosystem sort of form and function are affected, and food web dynamics. And here I've based a lot on the research that involves the use of stable isotope analyses, which we're able to carry out through our Stable Isotopes in Nature laboratory that I direct here at UNB. So the first one, let's look a little bit at winter. And whenever I sort of talk about the kind of winter research we do, I like to put this figure up. It's based on a paper that came out in 1998, and really, because different people have different sort of impressions about what they mean by winter. And for me, winter is a period that, especially from the salmon's perspective, starts immediately following the spawning in the fall, and really sort of includes that period from that point in the late fall or mid-fall, depending on the river, until the start of the smolt emigration in the spring. And during that period, if you look at that figure in front of you, you'll see two lines in that graph, and one represents the discharge regime and the other one temperature. And temperature, as you would expect, in that early winter period is declining rather steadily until, in many of our rivers, it reaches approximately zero, where after that it's pretty stable at about 0 degrees for much of the winter before increasing. And discharge also tends, in general, in many of our rivers to decline also, and reaching sometimes the near annual low during the winter, usually in about February. And the winter period is rather important for salmon because, especially if you think about the egg is really sort of a winter stage. It's laid sometime in October, November, and hatches sometime March, April, and then eventually emergence occurring sometime in sort of May or June, depending on the systems. So all kinds of things can happen: freezing, dewatering, acclimation to those temperature regimes, be it cooling in the fall or warming in the spring, all having different effects on different life stages of salmon. Especially if I think about direct winter influences, there are a number of here. Eggs are directly influenced by what's happening in the winter because of that period. Kelts, that post-spawning stage, salmon, unlike Pacific salmon, tend to overwinter, survive after spawning, and can survive to go out and come back to spawn again. So what do they do in the winter? Something we know very little about, and yet needs a lot of work. And the things that are happening in winter will very much affect how well the smolts will do and how many smolts we'll have coming out of some of these systems. So these are some of the areas that have sort of intrigued me and my colleagues over the years for looking at winter. So here's a slide showing different relationships as it relates to winter. Here I'm talking about winter discharge. So the amount of water that's flowing in a river in the winter, often in many of our rivers in an ice-clogged channel. And so how does winter discharge affect different stages of salmon? So this is some work that's just been recently published, some of it incorporating years of work. In fact, that first, the upper graph, which is looking at egg survival relative to winter discharge. And so this is discharge from that period just after spawning until and prior to the snowmelt freshet in the spring. And if you look at that, generally there's a good relationship here for all of the years, in this case about 10 or 11 years at Catamaran Brook, where you see about 41% of the variability in the data explained by, in terms of survival, explained by winter discharge. In other words, as winter discharge is increasing, the likelihood or egg survival tends to increase. And a similar sort of relationship occurs in terms of when we're looking at PAR. In the second graph, we're looking at density change here. So imagine if you're thinking about the PAR that are in the river. Well, we've been finding, for instance, that the change from the fall electrofishing numbers or density to the early summer or post-winter densities, that change in density is much higher, or in other words, there's a greater decline in density as you have lower discharge. In other words, as discharge is increasing, the change from fall densities to spring densities or early summer densities is sort of dependent on that relationship. More water, probably less impact, lower mortality. Whereas for smolts, surprisingly, in the lowermost figure, doesn't seem to be any obvious effect from the data that we've collected for all our years up around the Miramichi in terms of the total smolt number and the winter charges. That's something I'm still not quite sure how to explain that, except it could be a variety of factors, including emigration as older PAR prior to that smolt stage. Here's a point that just came up when the 95-96 point is sort of a different or an outlier point in that egg survival graph. And I just wanted to put this one in because here's a situation where we've measured the lowest ever egg survival in one particular winter, despite having a relatively high discharge. But this was a particular year when we had a midwinter ice breakup, a mechanical ice breakup that caused such significant scour in the system that we ended up having the lowest ever survival at about 7% survival. In other words, 7% of the eggs only survived, and that had severe implications for many of the fishes as well as the salmon that particular year. But that's a rather atypical event. In fact, in this slide, you can see in that top photograph, that's actually Catamaran Brook after that midwinter breakup in about late January of '95 when we had cold weather prior to that, significant ice buildup, and then a midwinter thaw that caused a severe mechanical breakup and scour of that kind of size of ice. And if you've seen Catamaran Brook in the spring, that's a surprising amount of ice that you've seen in that small channel. We're also doing work on other types of ice impacts. Tommy Lin-Ansari, who's a former doctoral student and postdoc who worked with me, has done a lot of work in winter and looked at surface ice experiments and the importance of surface ice as cover and the ability of juvenile salmon, for instance, to use it as a cover feature and to use different habitats, for instance, small substrates underneath that kind of cover in the winter. In other words, beneficial aspects of ice. Jean-Marc Roussel, a former postdoc of mine also, who was now working in France, has been looking at anchor ice and found again that anchor ice has had relatively benign effects on fish, especially salmon in many of our rivers, where they continue to use these areas fine, even with anchor ice over top of them, and they don't tend to emigrate from areas in many situations. And we're following this up with more egg survival work in terms of different kinds of ice and how it affects them. In other words, as you're in bigger rivers versus smaller or medium-sized rivers and headwater streams, what's the relative difference in terms of the potential impact of ice on egg survival? And I have a new graduate student who's going to be starting that up in the Miramichi in the coming year. All right, so let's switch away from winter to high temperature, or to other natural stressors, in this case high temperature. We already know, for instance, from some previous work and from the literature, that upper lethal limits for salmon typically are around 29 degrees centigrade, whereas physiological stress starts to be realized at about 23 degrees centigrade. And we've actually seen this ourselves in Miramichi, where aggregations will occur, territories are abandoned by juvenile salmon once they hit about that kind of temperature, whereas the optimal growth for survival for salmon is typically around 15 to 18 degrees. Not surprising, Atlantic salmon are a cool water fish, and so they don't really like those high temperatures. But we've experienced in the last 10 years, probably at least every second or third year, temperatures or midday temperatures, at least for 1 or 2 days, up above 25, 27. In fact, in 2010, we measured for the first time ever in the little southwest Miramichi a midday temperature of 30 degrees centigrade. This is way too warm for fish, and the concern is that this is likely to increase in frequency under a lot of the climate change scenarios and the models that are being that we're exposed to. So one of the problems is, we've got these great, beautiful rivers in our province, like the Miramichi, big, wide, glacial carved valleys, not much riparian cover. You can see the effect of ice on this kind of slide in this photograph, sort of pushing away most of that woody vegetation from the edges. And then you have this big, wide, shallow stream with a lot of boulders often exposed, which are great heat sinks. And so during a lot of days of summer low water and these shallow conditions and high temperatures, doesn't take much for these streams to warm up significantly. So what do the salmon do? So this is, I mentioned the 2010 high temperature event in the Little Southwest. This is a photograph underwater at one of these small spring seeps that come in along the Little Southwest Miramichi, where I took this underwater photograph. There's probably about 300 or 400 juvenile salmon, mostly 1+ and 2+ salmon par, that are aggregated at that small, cool water source. These are fish that are supposed to be, at this time of the year, territorial. And you can see it looks more like a hatchery sort of situation in terms of the numbers of fish, very high density, all trying to get into that cool temperature. Cindy Bro, who did her PhD with me a number of years ago, did a lot of work in trying to understand some of this, not only what's happening in terms of the aggregation in the field, but also in the lab, taking wild fish and seeing, well, if they seem to be stressed and are changing their behavior, are there physiological indicators that might explain this? And sure enough, with these 2+, or fish in their third summer of life, these juvenile salmon, here's a sort of a composite graph showing the physiological factors that we saw that certainly indicate stress. The green line represents oxygen consumption, and you can see at temperatures from 18 to 2, oxygen consumption is relatively stable, and then jumps up rather significantly between 22 and 24 and hits a plateau. And at that point, they basically hit oxygen consumption capacity, and they're switching to anaerobic metabolism, and hence the muscle lactate that you see also is jumping up between 22 and 24, and even blood lactate, which leaks out from the muscle afterwards into the blood at a slightly higher temperature, around between 24 and 26 and 28 degrees, is being elevated. And so really good indicators that there's a physiological stress that probably explains what we're seeing in the field in terms of the abandonment of territories. And you can see, interestingly enough, it's happening at around between 22 and 24, exactly that situation we saw in the field when the aggregations and the abandonment of territories occur around 23 to 24 degrees. So we've been following up on this particular research of high temperature stressors in the wild. And we've embarked on a study with some colleagues in Quebec, as well as other colleagues like Alan Curry here at UNB, excuse me, to look at how big a phenomenon this is over a larger stretch of the river. And so we had some aerial surveys done where we were looking at sort of temperature profiles, and this is sort of the kinds of figures produced from these airborne surveys with these thermal imagery that can show you cool water represented by that darker and purple line hugging one bank, flow is from left to right here, and so you can see a cool seep coming in from the top or from the bottom left of the screen, and how it sort of hugs that one bank as it's sort of deflected by the current, and how the fish then might use these different areas if we identify them along a long length of the river, which we've done in different sections. This is part of a 3-year research study in different rivers within New Brunswick, as well as in Quebec, looking mostly at Atlantic salmon responses. I have a graduate student, Emily Corey, who's doing her PhD to look at this, to look exactly at what's happening, how far fish are moving, where they're going, which kind of cool water refugia are more attractive than others, and how we might then best protect these areas and understand how the fish are finding them and what makes one refuge area better than another, perhaps. And we're doing this by tagging individual fish, monitoring their movements during high temperature stress events and in between, or when the stress is gone, and then logging these data on site around these antennal fields, as you can see in the top left photograph there, which is at the mouth of Otter Brook, one of these cool water refugia in the little southwest Miramichi. So, for instance, here's one figure that shows some of the results. Otter Brook is a cool water stream, a second-order stream that enters the little southwest Miramichi. You can see it there entering the stream. Again, runs from left to right here. The flow runs from left to right. This is the little southwest Miramichi. Otter Brook, you can see, is a slightly darker color in blue there, as it's cooler water entering the little southwest. Plume is discernible or measurable in terms of a temperature difference of 5 degrees for about 50 meters downstream. And we marked fish in spring prior to the warming up of temperatures at 5 locations, represented in numbers 1 to 5 there on both sides of the river, upstream and downstream of these seeps. And then there's another set of bank seeps, smaller seeps, at the very top of the figure up here, sampling site number 5, that you can see the blue dots along that upper bank or left river left bank. And so we marked the number of fish, approximately 40 fish at each of these locations, returned them to the site, and it came back at different points in the summer, including during one of these high temperature events that we saw in early July. And here's one situation where at Otter Brook, that particular day, that was the day that water temperatures hit about 30 degrees. We were able to track over that plume. There were probably, we estimated, anywhere from 4,000 to 5,000 salmon par aggregated at that particular cool water plume, of which we were able to detect 81 pit-tagged fish within that particular plume of cool water, whereas only about 2 pit-tagged fish were found on those seeps up at the top of the figure near site number 5. And in that pie chart that you see in the bottom right, it's basically representing, of those 81 pit-tagged fish, where did they come from? What were their origins? The pit tags tell us where they came from, and the numbers 1 to 5 represent the different sampling sites around Otter Brook. So you can see in that Otter Brook plume, the aggregation that was that was stressed by the high temperature, they came from anywhere from site number 1 upstream and the opposite side of the river, sites 2 to 4, or the same side of the river but downstream, sites 3 to 5. But more interestingly, we found fish from Catamaran Brook. 7% of those fish were from Catamaran Brook, which is 4 to 5 kilometers upstream. And Israelite, ISR site, 22% of those fish came over. That's about 3 kilometers upstream. And in that particular site, there are no immediate cool water refugia, so those fish had to move during a very stressful period to find these kind of refugia. We even had fish from the 2009 sampling the previous year at Otter Brook that were also located here. So it's giving us an idea now about how far fish are moving. We know, as far as we've been measuring, we've got some fish that have moved as far as 6 to 7 kilometers. These are small juvenile salmon par that are moving during some of these heat stress events. And this is the concern we have. Here's a figure of annual air temperature from 1900 to the present day, and you can see the pattern is one of increasing temperature, as we are all pretty familiar with now. And so this is going to be something to be very concerned about, especially when you consider that this is happening at least once every 2 to 3 years, and the salmon par in the Miramichi spends at least 3 years of its life before heading out to sea as a smolt. So it's very likely that it will encounter these kind of stress temperatures. All right, let's move on to anthropogenic stressors, human-induced stressors to rivers and how different fish are sort of reacting to these stressors. And especially let's look at forestry. This is some work we did for many years. Catamaran Brook was started in 1990, that project in the Miramichi, initially intended to look at forestry impacts, in this case clear cutting. That some of that work was published in a paper as part of a Fishes and Forestry book done by Northcote and Hartman back in 2003, I believe that, or 2004. And I had a chapter in that book along with some colleagues like Alan Curry and colleagues from Newfoundland, Dave Scruton and Keith Clark, in terms of looking at how fish reacted, especially salmon and brook trout, to forestry impacts. Basically what happened, this is the main effect of the forestry impact at Catamaran Brook, was really a hydrologic. We found very little and no significant biological effects from the kind of clear-cut harvesting that happened in Catamaran Brook over that period. And that was about, and that encompassed or involved about 8% clear cutting of the basin. The only effect that we saw was a hydrologic one. This is with my colleague Danielle Casey, and here's a figure redrawn from his 2002 paper that really shows in the lowermost figure that the timber harvest line, which is elevated from the all data and calibration line, in other words, that peak flows shown on the y-axis increased for any given precipitation event relative to the calibration or pre-harvest years. In other words, in Catamaran Brook, timber harvests seem to cause higher peak flows in the upper tributary only for a given precipitation event compared with the situation that it used to be in the years prior to timber harvest and compared with the whole river. And so only in that one tributary, that one subbasin, did we see a hydrologic impact that could be discerned as a result of timber harvest activities. And that one's not surprising because that particular subbasin, when you look at the amount of clear cutting that occurred, represented about 24% of that subbasin. And most of the literature and previous studies that have shown significant effects of forestry have all found that it happens when you get about 20% of a basin cut or a subbasin harvested. And that only occurred in that particular subbasin of Catamaran Brook. More recently, we've started to do a lot of work on looking at hydroelectric impact, or potential effects from hydroelectric operations. I'm part of an NSERC-funded research network called Hydronet. This is a nationwide network of representatives from industry, mostly Manitoba Hydro, BC Hydro, NB Power has recently joined this particular group. But you can see by the pins on the map that we have work, or we're doing work in as part of this network, or pretty well across the country from Newfoundland to BC, and including in New Brunswick. And the New Brunswick work, which is done largely through my lab and with some help from people from Quebec, is being focused on the Tobic system and looking at the impacts of hydroelectric operations on the salmon population and other fishes there. So the New Brunswick research, if I can sort of summarize, it involves, it's in my lab, 3 graduate studies, 2 master studies, one by Paula Toms, who's looking at how flow regulation affects salmon egg survival, and she's specifically interested in looking at water quality within the red, that hypereic water quality, and especially dissolved oxygen, and how it may be affected by flow regulation. The flow regulation in the Tobic system, she's mostly looking in the D and the Serpentine subbasins, is largely driven by flow changes and flow regulation from the storage ponds in the headwaters that are meant to sort of fill the lower reservoir behind the Tobic Narrows Dam, where the hydroelectric generation occurs, or where they release water for the Mactaquac downstream. There's a PhD study that's just started, and Adrian's looking at food webs and how they'll be affected using some stable isotope, as well as regular sampling to look at fish assemblage changes in unregulated versus regulated subbasins within the Tobic system. And finally, Sher Vu, who's another master student of mine who's in his second year now, is doing a largely semi-controlled experiment at Mactaquac using the outside channels to look at how hydropeaking or simulated hydropeaking flows, these would be daily fluctuating flows, affect the overwintering condition and lipid reserves of overwintering juvenile salmon, and especially how it might affect the ability of juvenile salmon to complete smoltification. So these are just sort of an overview of those 3 studies that are being carried out as part of the Hydronet research. And the nice thing about this particular work is not only is it addressing local things, but it's tied into a network for us and for our students to compare with students elsewhere across the country, how their results compare, and should help us all understand better what's happening in terms of hydroelectric impacts across the country. Here's an example for the D and the Serpentine rivers, and so on the y-axis you have the discharge measurements, in this case cubic meters per second, for the winter period from November until May, or the spring freshet. And if you just look at, I know you don't have different colors here, but the line, the smooth line, the darker black smooth line, is the line that represents the regular or typical hydrograph for northern New Brunswick rivers, and it's represented from the hydrograph from the Grand Riviere gauge station, and you can see low and some elevated discharge in November, December. This is based on the 2000 hydrograph. The regulated systems, and you can see very abrupt increases and decreases depending on the need for them to release water from the storage reservoirs. And so you see these very abrupt and then stepwise declines in the flow patterns. And so that's what we want to look at. You can see it's very different, and especially there's much higher winter flows than is typically the case in a natural hydrograph. Okay, the final area, or the last part I want to talk about, is looking at sort of these ecosystem, or what I'm calling food web dynamics. And here I'm sort of trying to understand sort of the place of salmon within this larger system. So I'm not just looking at salmon alone, but really what are the other biological or biotic components out there, how they're interacting with salmon, and even some of the abiotic features to help us understand the functioning of these systems. So one of the things we noticed when we came to Catamaran in the early 90s is there was an awful lot of beavers. And these beavers were building dams often in late summer and early fall, and then depending on the amount of water that was flowing in any particular year, were a barrier to some extent for the adults, or a complete barrier in these low water falls. 1994 is an example where basically we had a dam that was built about 500 meters upstream from the mouth of Catamaran, shown in that figure there, before it entered this little southwest Miramichi. And that was such a low water fall that as the adults came in, they only had about that ½ kilometer of river to spawn in, and they were unable to get over that barrier because there was no major rain event to breach the dam for any period of time to allow adults to get upstream. You know, compare that. So basically, as I indicate in that map figure, basically less than 1 kilometer spawning access. So the whole rest of the river that otherwise would be accessible was denied for those fish to get up into sort of into lay eggs. 1990, by contrast, we had about 10 kilometers of spawning access by adults. So this has all kinds of implication for population dynamics of these fish and the interactions with other species of fish. In a situation like in 1995, when we went to electrofish after the 94 low fall and reduced spawning access, of course, juvenile densities were very low in much of the brook. However, for the few fish that may have get certain years around some of these barriers, you'll have density-dependent effects depending on up above or below these particular barriers. So there's all kinds of interesting dynamics that can come as a result of these, again, a natural sort of component to the system, and in terms of how they interact with precipitation events in these areas as well. So we've been working on this, and here are a couple of examples of some papers that we've actually published. Sean Mitchell was a PhD student of mine in around 2004, 2005. And here's a paper that he published looking at this interaction of salmon and stream flow and beaver dams. And what he really found was very much the stream fish communities were very much affected by how well the salmon were able to access different sections of the river. And whether salmon accessors seemed to have a negative effect on other coexisting native species like slimy sculpin in these areas, and so very much could affect community and fish assemblages in different sections. And so these salmon, the beaver dams, and depending on the amount of stream flow in a particular year, all interact to sort of change and make these very dynamic systems. We also have found from some work by Doug Sigourney, who was a PhD student co-supervised by myself and Ben Letcher, and who did his work actually at UMass and Amherst, but worked at Catamaran, found that beaver activity and the areas above beaver dams actually were beneficial to growth, where growth of these salmon parr, H2 salmon parr, or pre-smolt year salmon parr, actually were increased, and their condition increased in these sort of inundated areas and riffles immediately upstream of these dams. So some interesting dynamics that we're starting to look at in this regard. So you'll see then, one of the things we do at Catamaran Brook, we've got a huge data set now where we have a lot of electrofishing data for different sections of the river for over 30 sites for now about 15 years, 16 years of data. Here's an example of some of that where we're looking at young of the year salmon densities at these sites in 3 different reaches of Catamaran Brook, represented by the 3 colors here of the lines in this graph. So the y-axis here represents density in terms of numbers of salmon per 100 square meters, and we've got the year along the x-axis. And you can see these are variables. So the middle, and then there's 3 reaches: the lower reach, or the lowermost section of the brook, the gorge reach about 4 kilometers upstream from the mouth, and the middle reach about 6 kilometers upstream. And whether or not fish can access, the adults can access certain areas will affect the summer densities of these 0+ fish, or the recruiting individuals from successful spawning the previous fall. So look at 1995, for instance. If you look at 1995, there's a sudden drop from 94 in terms of the gorge reach, and very low densities in the middle in 1995. Remember, 94 was the period of really low fall water. 0+ fish, or adults, weren't able to get up into the middle and gorge area, and the only spawning occurred in that lower stretch of the river, and so that's where densities are higher and very low in 95 in the other reaches upstream, until such years like in 97, where access was much better, and they could get up there because in 96 there was all kinds of water in the fall. So understanding these fluctuations, these dynamics, is not only driven by the number of fish that come in, but also by the interplay of a lot of these other components in the system to help explain how salmon are able to access different parts of the stream. So here's from Sean Mitchell's work, also looking at these different reaches, the lower reach, so going from the lowermost reach, and then upstream as you're going left to right, and then the percentage of the total production of fish that we find in these different reaches for different habitats. And then the different colors or hatching of the bars represent different species. You can see in the lower and gorge reach, it's pretty typical regardless of the habitat that salmon are the dominant fish until you get up into the middle reach, and then all of a sudden sculpin are much more known as our brook trout, and then until you get into the headwaters in the upper reach and in tributary 1, where salmon generally don't have access because of impassable barriers. That's where you'll see no salmon there, and instead you'll see much greater numbers of brook trout and dace, as well as sculpin. And so, and that, especially in that middle reach where salmon can access in some years, that representation of what's actually occurring there in terms of species richness will very much be driven by other factors like access to previous years and beaver dam interactions. And finally, the last thing you do, there's a fair amount of work that we're doing looking at ecosystem function in food webs by looking at stable isotopes. And stable isotopes is a wonderful that ecologists have sort of picked up on in the last 20 years. So to help us understand energy flow systems and diets and access of different habitats depending on the isotopes we use. Typically we use carbon and nitrogen. This is a typical biplot of carbon, nitrogen, carbon on the x-axis, nitrogen on the y-axis. And nitrogen is usually a good indicator of trophic level, whereas carbon is a good indicator of food source. And all this is meant to show is this is some work from Tim Jardine, who worked with me many years ago, where we were looking at marine-derived nutrients that might be coming in as a result of anadromous fishes, whether it's salmon, in this case, alewives, represented by alosa, the point's called alosa there, which is up in the top right of each of the two graphs. And here, that's the signature, the isotopic signature in carbon and nitrogen for this marine fish that come in large numbers, excrete, lay eggs, sometimes die, excrete these nutrients, and how are they taken up by the freshwater system and the components in the food web was our question. And all I want to point out to you here is if you were a completely marine signature like the alewife, you would have a point which is represented or overlaps with the alosa points in the upper right of each of the two figures. What you're seeing here are invertebrates. In the upper panel, you're looking at Dronella, which is a mayfly, and in the bottom one, you're looking at flies. And you're seeing here these, my fellows. So you're looking at, here's the alosa, the marine signature, represented here. And if we would expect that there's nutrients coming in by these marine organisms, then they should start reflecting in their signatures something similar to this. Now, in these particular streams, in this case, this is from West River near Pictou, Nova Scotia. The triangle points here represent different individuals of dragonflies from a point upstream of a barrier where alosa cannot access. The circles here represent those within that area downstream of the barrier where alosa can actually, and periods of sampling, but the point is it's the same species, only separated by a barrier between the and you can see stream points, especially the ones that came in after alosa spawned, which were the colored circles here, are showing signatures that are getting closer to that of a marine signature. In fact, they're these marine nutrients either eating the eggs directly or picking up the salts components excreted by these fish.
Welcome to GoToWebinar, Web Events Made Easy.
And are concerned to us because our numbers are down from what they were historically. And so what's the potential effect of these fewer or this less of a marine influence and how that is affecting freshwater production? And that's basically of the salmon-based research that we're doing in Catamaran. This, of course, is, for those of you who've been there, a lovely photograph of the Little Southwest right at Catamaran Brook that we like to look at and spend as much time as possible on. So thanks for your attention. I'd be happy to take any questions you might have.
Okay, so people, there's a couple questions that were asked during it, but if other people want to raise their hand, you can get in the question queue. So the first person I'm going to unmute is Peter Salonius. He made a comment earlier in the talk about anthropogenic impacts. Okay, so Peter, if you want to go ahead and speak, you're unmuted. Oh, maybe we've lost Peter. Peter Salonius, are you there? He had made a comment just about asking about anthropogenic impacts, saltwater, sea cage, origin, sea lice. So I don't know if it was more of a comment. Okay, I'm going to mute Peter, and then we have Denny Isaac, who just has a question about temperature. Just a sec, Denny. Okay, you're unmuted. Let's get Denny.
Hi.
Hi
Can you hear me?
there.
Yes.
Okay, yes, I was asking a quick question about your the effects of temperature on the survival of species in the rivers, and I was wondering if you had any more information on, say, trout or American eel or any other freshwater species.
Well, we certainly see that the trout have even less tolerance of the kind of temperatures that you saw me showing for the Atlantic salmon. So they tend to be have an even lower tolerance for that, and so we're seeing them certainly being affected earlier than the salmon in these rivers. So in the Little Southwest, or in much of the Miramichi, for instance, the brook trout vacate these main big river corridors, like the one you're looking at on the screen right now, the last slide, and they tend to go up into the tributaries much sooner than the salmon ever start to react to. So yes, we're certainly seeing, and we're also seeing effects of species like slimy sculpin, another cool water fish lower part of Catamaran Brook and much of this part of the little southwest Miramichi, for instance, is too warm. The only place we see slimy sculpin are in the cooler tributaries or in the upper sections where the water temperature is within their range.
Thank you.
Okay, next I'm going to unmute Joshua Roy.
Hello, Rick.
Hi.
There we go. I'm wondering, with the thermographic imagery that you did, what method you used, some relative costs compared to getting a bunch of grad students out there with digital thermometers and canoes or hiking stream banks. It just seems like such a efficient way to cover a lot of ground, but maybe not so much if you need a helicopter and
yeah, these were fixed wing surveys, so they were very expensive. And there's I know in our case, I don't know the exact cost, but I know it was rather expensive. First of all, you've got fixed wing aircraft, and then we brought in the people that had to run the software and had the particular equipment. They came from the west coast of North America, from Oregon, I believe. We now have people in Quebec who can do this, but and we did try it a little bit cheaper way of doing it by these remote controlled, almost like drones with the fixed cameras on them, but it didn't work nearly as well as the fixed aircraft, manned aircraft imagery. But
because of the steadiness of the drone,
you can cover a lot more area. But you know, you can understand, or if you know river systems well enough, or spend a fair amount of time either in the middle of the summer or in the winter, you can also find a lot of these refugia pretty well, or most people know of them just by walking along riverbanks. If even nothing else in sandals, you pretty quickly pick up which is the cool water sources, and or in the winter when you see areas that don't freeze as quick, they're usually indicative of a groundwater discharge of some sort, and so that sometimes can help do it in a much cheaper way than with the aircraft.
Excellent. Do you have time for another question?
Yeah, go ahead.
It's about Catamaran Brook and the harvesting. I'm wondering if, the proximity of cutting, you mentioned that only about 8% of the watershed had been cut, and that studies show that you really don't see impacts until you approach 20%, but I assume that has everything to do with the proximity of the cutting to the brook and tributaries, and if they're cutting forested wetlands that might be source areas for water.
Absolutely. In the case of Catamaran Brook, we decided at the very beginning that we weren't going to dictate where to harvest. Rather, we were asking both the province and the forestry company to harvest as per their timber management plans. So they use the regular plans and restrictions as would be required anywhere else in the province. The idea there being not that we wanted to so much see an effect as we wanted more to see if cutting as is done typically in the province would have an effect. So at two of the harvest blocks in the lower part of the system, the riparian buffers were 60 meters, and in the headwater smaller streams it was 30 meters. So you're absolutely correct. If there were no buffer strips, the likelihood of realizing effect would have been much higher. If we didn't have adjacency rules to harvest blocks or even harvest block sizes, we probably would have seen an effect. But even in a case where we did have it, we did see certainly an elevated turbidity event because of an improperly sized culvert during a high rain event and harvesting probably when they shouldn't on a rather steep slope. But even with that, the actual measurement of biological impact, we were unable to detect it. Probably partly indicating the relative resilience of natural systems, but also the fact that it was a small area and its impact on a population level was could only happen if you really hammer the system hard. So anyway, that's what we found.
Yeah, good. Thank you.
You're welcome.
Okay, we're going to try Peter Salonius again. Just hang on, Peter. Let me find you in the list here. Okay. Okay, you're unmuted, Peter. Go ahead. Oh. I don't think his mic is working. Peter, if you have a question, do you want to try and type it? because it doesn't appear that we're not hearing you.
And Peter, I mean, or you can call me later, but if the question has to do with, I think there was some reference to lice, I presume you must mean from some of the cage salmon in the Bay of Fundy. I'm unaware of any of the work that's being done in that regard, although I know it's a concern for a lot of people, but I actually don't have any direct information on it.
Okay. I think Peter, you're self-muted right now. I don't know if you can unmute yourself because I've unmuted you. Oh, there you go, Peter. No, we're still not hearing you. It might be a mic problem. There's always glitches with technology. Well, I think we've gone a little bit over time than we planned, so I'll just wrap things up and thank everyone for attending, and also thank Rick for the overview. There was a lot of topics that were covered there. We will have a PDF of his presentation and a recording of this webinar put on the Atlantic Salmon Conservation Foundation website under resources. That will be within the next day or two. I just want to remind everyone that the next webinar in this series will be on February 13th. David LeBlanc of the Restigouche River Watershed Management Council will be speaking about habitat assessment, and this presentation will be in French, but there will be a bilingual question and answer session following. After that, the next presentation that will be in English will be Todd Dupuis of the Atlantic Salmon Federation and Fred Parsons of IRMA. They'll be speaking on some projects in PEI, and David LeBlanc, I think my notes are messed up a bit. Anyways, we've all the webinar listing is on CRI website as well as ASCF. So registration for all the upcoming webinars are available on the Atlantic Salmon Conservation Foundation website under resources as well. So thanks everyone for participating.